Well, hi, everybody. Thanks for sticking through several hours on Zoom and for being here today. It's really good to see everybody again, quote unquote. My name is Katie Lowe. I'm a master's student working in the Haney Lab at UNH. And today I'm going to be sharing some preliminary results from an exploratory study looking at anatoxin accumulation in Yellowstone food webs. So you've already heard quite a bit about anatoxins through the previous talks, so I'm not going to go into really too much background detail here, but I do want to highlight some important characteristics of anatoxins as they relate to my work dealing with food webs. So as Haley mentioned, anatoxin is a potent neurotoxin produced by several groups of cyanobacteria. One of the main ones is Dolichospermum. Anatoxins have been named very fast death factor because they take effect very quickly. Within just a few hours of exposure, you may see neurodegenerative effects, including loss of coordination, muscle twitching, or you might even see respiratory paralysis and death. And so as a result of this, anatoxins have garnered a lot of attention in the news lately, especially following the deaths of several dogs that have come into contact with harmful cyanobacteria blooms. But despite the severe health risks associated with anatoxins, most of our cyanotoxin research focuses on microcystins or other toxins. And so we do still have quite a bit to learn about how anatoxins move through the environment. And anatoxins are reported to degrade very quickly, as you've heard. They're also highly water soluble, so we don't really think of them as a toxin that would accumulate in the environment. But there are studies that are based on fish models primarily that show that anatoxin depurates from the body very quickly, but there are other studies that show that anatoxin does accumulate in both aquatic plant and animal tissues. And potential accumulation in biotic tissues could significantly increase the risk of dietary anatoxin exposure to consumer species. And we know that ingestion is an important exposure route of anatoxins. Anatoxins are rapidly absorbed into the blood via the gastrointestinal tract. But in order to better quantify the risk of dietary exposure to anatoxins, we first need to have a better understanding of how they actually move through food webs. The studies that have looked at anatoxin accumulation in animal tissues have primarily focused on fish. And so at this point, it's really unclear if anatoxins are accumulating to a significant degree in other compartments of the food web, such as macroinvertebrates, or how the degree of accumulation might differ among species that occupy different positions in the food web. And typically the biggest concern that we have with toxins in food webs is whether or not they're biomagnifying or becoming more concentrated as we move up the food web from the level of primary producers to apex predators. And that's because if this concentration, if this biomagnification is occurring, species that feed high in the food chain, including humans, might be exposed to relatively high toxin concentrations, even when those background concentrations are very low. And so in addition to knowing whether or not anatoxins are accumulating in consumer tissues, we also want to know a little bit about this pattern of accumulation across the food web. So in order to answer these questions, I used samples that were originally collected as part of my master's research, which is looking at the trophic transfer of BMAA in food webs in the Greater Yellowstone ecosystem, which is the area in and around Yellowstone National Park, shown by the green line here on this figure. And for my master's work, I sampled a total of 12 lakes in this area, but most of my sampling focused on these two systems. You can see they were both located right along the southern boundary of Yellowstone National Park, marked by stars here in the figure. And these two lakes were very similar in size, location, and elevation. They were both relatively nutrient poor. They were expected to support relatively low primary productivity, but they did differ in their food web structure in that one contained small planktivorous minnows, while the other was entirely fishless, as are about 70% of Yellowstone's lakes. And so this gave us a really unique opportunity to be able to determine how anatoxins are moving through food webs that differ considerably in their structure. So at each of these two lakes, we sampled surface water via an integrated tube sampler. We also collected net phytoplankton and net zooplankton with plankton tow nets. We then collected littoral phytoplankton from nearshore areas, as well as benthic phytoplankton from the sediment water interface of sediment cores taken from 3 haphazardly chosen locations at each site. And then from these samples, we measured concentrations of the photosynthetic pigments chlorophyll, phycocyanin, and phycoerythrin, so that we could estimate biomass of all phytoplankton as well as cyanobacteria specifically. And then we measured toxin concentrations using ELISA analysis. And so just to give you an idea of what we found in this lake, when we were looking at the water column itself, we found an abundance of picocyanobacteria. The majority of those picocyanobacteria were non-colonized, but we did find Aphanocapsa as well as Chorococcus in these systems. You can see that the primary productivity in the water column was fairly low, especially for PC. We found that phycocyanin concentrations were really right at the detection limit of our handheld fluorometer. And so as a result, we found a very low toxin concentration. But when we looked at the nearshore areas of these lakes, we saw that the amount of primary production significantly increased. We saw a shift in the phytoplankton community toward diatoms and also slightly toward Dilithiospermum. Again, this is one of the major producers of anatoxin. But despite this shift, we didn't see an associated increase in toxin concentration. And so as a result, the toxicity of our samples, which is measured by the amount of toxin per unit biomass, as estimated by either phycocyanin or phycoerythrin, was significantly lower than it was in our pelagic community. And we found the same trend to be true when we looked at the benthos. Again, we saw a big increase in pigment concentrations, another shift in the community composition. The benthos had a lot of diatoms as well, but for Fishless Lake in particular, we found a pretty thick benthic mat of oscillatoria. But again, we didn't find an associated increase in toxin concentrations, and so our overall toxicity was very low. I will mention though that this was really only true for anatoxin. The original work that I did on this on these lake systems with BMAA did not follow the same pattern. We did see differences in toxin concentrations across these 3 different sites within each lake. And I will also note that the anatoxin results that I'm presenting today are based on a single replicate sample from each of our lake systems. So while these do give us an idea of whether or not anatoxin is present, they don't give us a good idea of what the variation in anatoxin concentrations might be. So in addition to the plankton sampling that we did, we also sampled macroinvertebrates, frogs, fish when they were present, and common loons from each of these lake systems so that we could determine if and how anatoxins were accumulating in consumer tissues. We again measured toxin concentrations using the ELISA analysis, and we also used stable isotope analysis to quantitatively identify the trophic positions, or the position along the food chain of each organism that we sampled. And so this was basically a way to kind of try to define our food web a little bit better before we looked at how anatoxins were moving through it. And we found that anatoxin does accumulate in these consumer tissues. We detected it in 21 out of the 22 consumer tissues that we sampled. We found that anatoxin concentrations in the consumers, which are represented by these samples, were significantly higher than those in the producers. But we did find pretty high variability when we compared anatoxin concentration across the different consumer groups. These values are averaged between the 2 lakes, but you can see that across the consumer groups, values range from about 7 to about 400 nanograms per gram. And just to give you an idea of how these values compare to the couple of studies that have been done on anatoxin accumulation in tissues, we see that they do fall within the range of those reported for a study that was done on a eutrophic reservoir in Poland. They found the range of anatoxin concentrations in fish muscle tissue is represented by this gray band, and the average was represented by this black line. So again, you can see that the values that we're finding in these organisms are pretty similar to the values reported in other studies, although they do fall below that average value. That's not terribly surprising. Again, these lakes that I'm studying are oligotrophic or meso-oligotrophic. fairly nutrient poor. We didn't find any evidence of a cyanobacteria bloom on them, and they are in Yellowstone. So we really don't expect toxin concentrations to be high. And the study that we're comparing to was looking at a eutrophic reservoir with a cyanobacteria bloom. So now that we know that anatoxin is accumulating in these consumer tissues, that accumulation does vary among the consumer groups, we next want to know a little bit about the pattern. Again, we use stable isotopes to determine where these different consumers fall in our food chain. What is their trophic position? And if anatoxin is biomagnifying, we would expect that the toxin concentration would increase either linearly or perhaps exponentially as we increase in trophic position from primary producers to apex predators. We used, again, stable isotopes, and we calculated trophic position following this equation. And when we look at these regression results, we see that anatoxin does appear to be biomagnifying. It appears to be increasing in toxin concentration, but only up to a certain point. And then once we transition into the higher trophic levels, higher trophic positions rather, we see that there is a decline in anatoxin concentrations. You can see that this relationship is significant, but there is still quite a bit of scatter in these data. And that's likely due to relatively low sample size combined with the fact that we are looking at data from two different lakes with known differences in food web structure. But despite these limitations, we found that this trend is very similar to what we were observing with BMAA in these systems. So again, this is looking at the relationship between trophic position and toxin concentration. With BMAA, we had a slightly larger sample size, and so we were able to split up lakes with and without fish, but we saw the same trend in both cases. You can still see that there's quite a bit of scatter in this relationship. We're still combining multiple food chains, and I do still have some work to further tease that apart. But one of the main takeaways that I want to share with you guys today is that really regardless of which lake we were looking at, which food chain structure, or which toxin we were looking at, we pretty consistently found that macroinvertebrates were accumulating some of the highest toxin concentrations in these systems. And so this could mean that anything that's consuming those invertebrates would be at a higher risk of the negative health effects associated with that cyanotoxin exposure. And this is particularly important because several of the macroinvertebrate groups that we sampled are emergent. So as adults, they leave the lake system, they fly away, and they take their toxins with them potentially to terrestrial systems. So briefly, I just want to touch on the risk associated with the accumulation that we're seeing. As has been shared in various talks throughout this mini conference, we don't really have a good understanding of the risk associated with anatoxin exposure. The information that we have is not sufficient for developing reference values. And the current risk models that we have really largely focus on death, on very acute effects. We know very little about sublethal effects, and we know very little about the chronic effects of anatoxin. And when we talk about this from an ecological perspective, a lot of these sublethal effects are going to have potentially important ramifications for the overall health of wildlife populations. They might be affecting behavior, they might be affecting growth or perhaps fitness. And so these are things that we should be accounting for when we talk about risk. We should also note that we did find multiple toxins within these food webs. The preliminary data that I've shared today shows that these toxins appear to be accumulating in similar patterns throughout these food webs, which means that they appear to be accumulating in the same organisms. And so we should try to understand how these toxins might be interacting with one another, whether that's an additive or synergistic effect. We also know that that mercury levels in this area tend to be high. This is Yellowstone. There's a lot of volcanic activity in the area. And so although we don't have numbers for this yet, we do know that mercury levels do tend to be high. And so again, that's another neurotoxin added into this system that might be having an effect that we aren't currently accounting for as it interacts with these other neurotoxins. So just to briefly summarize, we found that anatoxin levels were detectable in these Yellowstone lakes. This is not really what we expected. We expect Yellowstone to be a pretty pristine environment, but we are starting to see more and more cyanobacteria blooms in this area. And as I've shared, we are seeing benthic cyanobacteria mats forming in this area as well. Although the background levels that we found for anatoxin were fairly low, we did find that anatoxin is accumulating in consumer tissues at significantly higher concentrations than are those background levels. We found anatoxin in every food web compartment that we looked at, which really stresses the need to consider more than just fish when we talk about accumulation models. We also found that the pattern for accumulation of anatoxin as well as BMAA are not linear in these systems. They didn't follow that linear or exponential trend that we associate with biomagnification. And so while we still don't really understand why this sort of curved pattern is occurring, it is suggesting that macroinvertebrates appear to be accumulating more toxin than just about any other species in these systems. And that could be an important exposure risk for species that feed on those macroinvertebrates, including consumers in both aquatic and terrestrial systems. And we do still have a lot to learn about trophic accumulation of cyanotoxins. We need to know a lot more about the risk assessments, develop those models further, and we need to know about potential additive or synergistic effects of multiple toxins, considering both chronic and sublethal effects in addition to those acute severe effects. Ultimately, these results highlight the need for more research. They emphasize that monitoring should include nutrient poor lakes and systems that may have multiple toxins. Our results also highlight need for additional research into the effects of these toxins, particularly when those multiple toxins are present. So with that, I will just quickly say thank you to everyone who has helped me with either collecting or processing these samples. There have been a lot of people involved, and I look forward to taking any questions that you might have in the breakout rooms. Thank you very much.